Thank you very much. I want to thank everyone for being here this afternoon. It's a wonderful location. I'm going to tell a story about some new science, if I can do this properly.
Go the other direction.
Yeah, got it now. Got to pass the thank you. About a target that applies initially to ALS, but it's going to be, by the end of the talk, will be applicable to Alzheimer's disease and frontotemporal dementia as well. And it's about a pathway, an S-core pathway known as, that we focus on, CHMP7. Unfortunately, Dr. Coyne, who was going to do the presentation, is ill today, so I'm doing it instead of her. But we could have easily both stood in front of you. I'm a neurologist and neuroscientist. I run the Brain Science Institute at Johns Hopkins and founded and run the ALS clinic. The focus of this initial therapy will be Lou Gehrig's disease, or amyotrophic lateral sclerosis. This is one of the worst adult diseases imaginable, other than some of the cancers represented in this audience. It affects maybe about 30,000 Americans per year, probably more, several hundred thousand worldwide. And quite frankly, if we could keep patients alive longer, it would be many hundreds of thousands. It's not an uncommon neurodegenerative disease. In fact, I guarantee each one of you will probably have a friend, neighbor, someone you'll know that will develop and die of this disease. 90% of the disease is sporadic. A small subset are inherited, usually in an autosomal dominant fashion. And we know a lot about the genes. We know very little, really, about the causes of sporadic ALS, and I'll get to that in just a few minutes. We do have therapies for ALS, 2 approved therapies, riluzole and darvone. Riluzole actually came out of my lab more than 20 years ago. We have very effective gene therapies. Closadi, or tofersen as I know it by its generic name, is a remarkable effective drug because it gets at the starting point of ALS. The vast majorities in ALS, as the case would apply to Alzheimer's and frontotemporal dementia, fail because they work at downstream pathways. I'm going to tell you about a pathway that's far upstream, analogous to, if you will, Closadi, and the ongoing drug trials with FUS, the FUS drug trial. It's believed, and I'm no expert on the market, it's believed this is certainly a growing market because the thought is that some of the drug candidates for ALS will certainly overlap. A fundamental pathogenic cascade in ALS is a protein known as TDP-43. It's a nuclear protein, and in the course of disease, it becomes cytoplasmic, and then eventually it aggregates. We know this from animal models, and we know the end-stage aggregation loss of nuclear from pathology studies. When it's no longer in the nucleus, it no longer has its normal RNA metabolism regulation that it normally controls, and this leads to thousands of dysregulated and aberrant RNA species that in fact lead to some cryptic, if you will, aberrant proteins. Prolonged aggregation in the cytoplasm is believed to cause neurodegeneration, but it's the loss of function, we're going to talk about loss and gain a lot here, that is the focus, and it's been the focus of some pharmaceutical companies. We'll come to that. So initially, there's cytoplasmic mislocalization of TDP-43. This applies to essentially all sporadic ALS patients, at least at postmortem. And when it's not in the nucleus, you have loss of function. Individual pharma companies, some may be represented in this audience today, are focusing on one of those many thousands of misregulated species. Curalis focusing on stathmin and Trace Pharmaceuticals focusing on UNC13. Interesting. I hope they work. I'm running those trials at Hopkins right now. Likely they'll fail because they're only fixing one of thousands. The other part of that equation is the gain of function, these aggregates. And there are several companies working on aggregates. The excellent science, DuPont does excellent science. However, none of them have ever shown they really repair the loss of function that occurs. They're all using very artificial models. We use a model that actually is truly sporadic ALS. Our model is completely based on using iPS cells, induced pluripotent cells derived from our patients. Those of you who study cancer, this is the equivalent of a biopsy-like generation from patients' blood cells, make iPS cells. I run a program called Answer ALS where we did this from 1,000 patients around the United States. We have essentially a large library of these cells, and they recapitulate the natural loss of TDP-43 function. There is no mouse model that does this, no other model that does this. So it's the perfect platform to then do your drug discovery and optimize your pathogenic cascade. Now, you might argue a model lacks something because it's not a model. We all know how models can have shortcomings. Fortunately, we actually have brain tissue from the same patients we generated our iPS cells. And the cascade of loss of TDP-43 function that we find in these iPS models, we find exactly the same phenotype in the brains of the same patients. So it's a pretty good model for at least this major pathogenic cascade in the disease. Well, what's upstream of that? I've told you what happens. What's causing the TDP-43 loss of function in the nucleus, again, common to all sporadic ALS, is we discovered a number of years ago, Dr. Coyne and I, that upstream of that is an injury to the nuclear pore complex. Nuclear pore complexes regulate the input and output of the nucleus, RNA coming in, RNA coming out, proteins coming in. And TDP-43 critically must get into the nucleus. It's normally nuclear. The injury to the nuclear pore complex that we've studied in patients' iPS cells, as well as brain tissue at autopsy, is defective in sporadic ALS. There's a loss of proteins that make up that complex, and that leads to TDP-43 loss of function in those living cells. As best as we can tell from our model, this is the initiating event that underlies TDP-43 loss of function, the major defect in sporadic ALS. By the way, that same loss of defect is found in half of Alzheimer's disease and a large percentage of frontotemporal dementia. So the biology we're discovering here and the target will be, we believe, will be very relevant to those diseases as well. We validated this in 100 living IPS lines. That's like a clinical trial. By having that kinds of large, it's not a typical lab experiment for my academic colleagues of 2 or 3 cells, it's 100 patient lines. And what's upstream of that we discovered is a pathway, an escort pathway known as CHMP7, in this case, a protein that normally modulates and regulates the nuclear membrane and nuclear pores. It's aberrantly localized within the nucleus. It's normally largely cytoplasmic. It's localized to the nucleus, causes nuclear pore damage, which downstream leads to loss of TDP-43 function. So the cell model can give us the temporal cascade and teach us that this is the upstream target. How do we know that? Well, we've developed antisense oligonucleotides, and we have a lot of experience in neurologic disease using antisense oligonucleotides. We're already with an FDA-approved drug in multiple clinical trials that we've run. For example, at Johns Hopkins, we can readily use them. These ASOs are highly effective and at a relatively low inhibition of the target. In this case, if we knock down CHMP7, which as far as we can tell have been quite safe both in animals and human cell lines. This is a busy slide, but what you can see here is that each row is individual patients, about 30 patients shown here. One column are the various species of TDP-43 loss of function. You'll see it's an alternating sort of a mix of red and white to full blue. That means we're completely repairing all of these TDP-43 loss of function species in our patient models. It's the only and perfect model to study sporadic ALS, and the drugs work very effectively, the ASOs, in repairing that injury. And therefore, we believe CHMP7 ASO has great promise as a therapy for ALS, and we think eventually for other TDP-43 apathies. And then instead of repairing just 1 or 2, Stathman or UNC13 as the existing companies, we're repairing all of them, which makes it a high, more tractable candidate, we think, for patient trials. And I almost lost those slides. And so sort of what we know is that in the nuclear pore, normally CHMP7 is in the cytoplasm, TDP-43 is in the nucleus. It relocalizes to the nucleus, damages the nuclear pore complex, and leads to inappropriate cytoplasmic accumulation of TDP-43, and then later aggregation of TDP-43. So CHMP7, therefore, is a novel disease-modifying target for the treatment of ALS. The CHMP7 ASOs, and again, I want to underscore, ASOs are readily usable in clinical trials. They serve not just as an effective therapy, it's a quick readout, it's a quick readout for biomarkers, for efficacy, for target engagement. We've been doing this now for almost a decade in neurologic diseases. It corrects the nuclear pore injury, and most importantly, corrects the defects of downstream loss of function that we see in ALS. So in summary, we know that nuclear pore injury precedes TDP-43 loss of function. CHMP7 knockdown with our ASOs, again in animals and iPS cells, can repair TDP-43 loss of function. And because of time limitations, we know that another member of the escort pathway, CHMP2B, works just as well as CHMP7, and it's an upcoming target for us as well for ASO development. I should point out we have other reagents beyond ASOs, including siRNA and the equivalent of PROTAC to get rid of this protein. And therefore, targeting TDP-43 upstream offers a really comprehensive way of thinking about targeting TDP-43 loss of function in sporadic ALS, but because the same pathophysiology is present in AD and FTD, we believe we'll eventually be able to move on with our model systems to apply this biology and therapeutic intervention into those other neurodegenerative diseases. We've been working with Blackbird Labs to expand this mechanism of action biology, and the slide sort of outlines the various things that we're going to be doing with them in terms of various developing some of the assays that we'll be using both for validation but also for eventually for our clinical trials, and enabling targeting such as CHMP2B. Thank you.